Okay, thank you very much, Klaus. Thank you very much for that kind introduction. And I'd like to thank you for that. I'd like to thank Shankai, one of your students, for hosting me as well, and for Julia for setting this up. So today what I want to talk to you about is work that's been ongoing in my lab, trying to understand the geographic spread of crops. And let's see now if I can. And crops have been, I've studied many plant species over the last 25 or 30 years. But actually in the last 10 years, I've really, or maybe 10, 15 years now, I've really focused my attention on the study of crop domestication as a way to understand the evolutionary process. And domesticated crops, as you know, are an interesting group of species. I understand you had Jeff Ross Ibarra and Brandon Gott give a talk there in the last few weeks, so you are familiar with domesticated crops. As an evolutionary model system, they're interesting because these are species that we think have evolved in the last 12,000 years, primarily as a result of a mutualism between humans and a variety of wild species that were the primary food source of many of our hunter-gatherer ancestors. But this mutualism, which began when human behavioral ecology changed, such that we moved from being hunter-gatherers to being herders and farmers as a way to get the nutritional resources we needed for survival. And because of this mutualism, these group of species, of which the numbers range from 200 to 2,000 plant domesticated species, have evolved in relationship to humans and under the selection pressure of this new environment, the environment which we now call the farm or the garden or the orchard, and in response to this human interaction. And as many of you know, there's a long history of the study of domesticated species as a way to understand the evolutionary process. Darwin famously, in the introduction of Origin of Species in the first chapter, discusses the use of domesticated crops, both as a way to study variation in populations and also the creative force associated with selection. And the first book, in fact, he published after Origin of Species, which was supposed to be the first chapter of his magnum opus, was the variation of animals in plants under domestication. And so since Darwin's time and earlier than that, and to this day, evolutionary biologists have used domesticated crop species, hopefully as a way to understand how evolution proceeds. Now, all domesticated plant crop species that we rely on these days for food have evolved only in the last 12,000 years. And they evolved in 12,000 years around the world in different places. So this map shows you where some of the major areas of crop domestication occurred, and there are about 24 regions where there have been multiple crop species that have evolved. The earliest example of domestication in plants was found in the Fertile Crescent, this region that goes from Iraq to the southern Turkish mountains into the Levant. And this is also where agriculture first started, starting about 11,500 years ago. This is an area where wheat and barley and flax and peas and lentils were domesticated, as well as sheep and goats and cattle. So this was an area where humans really began that first exploration of changing their ecology, of how to deal with gathering, getting their nutritional resources. But we know that domestication actually occurred in various parts of the world, and agriculture was established in different parts of the world independently from the Fertile Crescent here in the Middle East. We have in China the domestication of things like millets, which occurred independently from domestication of plants in the Fertile Crescent. And of course, in the New World, in the Americas, there was domestication of corn and squash and beans and sunflowers and potatoes, which again occurred independently from what was happening in other parts of the world. And of course, in Africa, Papua New Guinea, there are other places where domestication of plants has occurred. It's still not clear why humans changed their behavior so that they relied on farming and agriculture as a way to gather their nutritional resources. That's still a matter of debate. But what we know is it was a global evolutionary process that took place in different parts of the world. So my lab has studied different types of domesticated species as a way to understand evolution. There are two main groups of species that we are primarily working on in the lab. One is Asian and African rice, Oryza sativa and Oryza glaberrima. The other is Phoenix dactylifera, or date palms. And today what I'd like to do is talk to you a little bit about both these species. Let's start off with rice as an evolutionary model system. So rice is Oryza sativa, Asian rice. It is the most important food crop in the world. It's a primary calorie source for about half the world's population. Its genome is, relatively moderately sized, about 400 megabases. And there are a large number of varieties in rice. There are about 130,000 varieties that are maintained by the International Rice Research Institute, gathered from different places around the world. And Oryza sativa, domesticated Asian rice, evolved from, we believe, from Oryza rufipogon, or was domesticated initially from Oryza rufipogon, starting about 8 to 9,000 years ago in China. Now, domestication in Oryza did not only give rise to Oryza sativa, but in Africa, there was a separate domestication event of Oryza. Oryza glaberrima, or what we now know as African rice, evolved from Oryza barthii about 3,000 years ago. So these were two separate domestication events. And there's actually archaeological evidence to suggest that maybe in the Americas, there was also a separate domestication event for Oryza. But the one we're focusing on now is Asian rice, or Oryza sativa. And as I said, there's a lot of variation in domesticated rice. And one of the early things we did was we looked at sequence data for about 110 gene fragments across the genome. And we used that to look at the relationships between different rice land races. Now, our studies usually focus on land races, which are traditional varieties that farmers use. So these are not the result of scientific breeding. These are traditional varieties, and so we think are much more a better reflection of the evolutionary processes leading to the evolution of crop species. And when we do, a gene clustering or a phylogeny of these different rice land races, and we have from different groups of rice, as well as the wild species, Oryza rufipogon, and some outgroups, Oryza meridionalis and Oryza barthii, we find there are two major genetic groupings of rice, like kind of super groupings. One is a group of rice called the Indica and Aush. And Indica and Aush are thought to be subspecies of rice. And the other is Japonica and aromatic rice. So Japonica rice is the rice you would associate with sushi, for example. And aromatic rice is the rice that you would associate with like basmati rice of India. And each of these groups, Indica, Aush, Japonica, especially, have been thought of to be subspecies of Oryza sativa. And the original domestication of rice, the evolution of rice, has been controversial for the last 20 years. It has been thought that it evolved or domesticated at least 2, some say even 3 times. I'm not going to go into both the controversy and what we think is the answer to the paradox of the domestication of rice. I'm going to go through like a summary of several of over a decade of work. It's believed that Oryza rufipogon, which is the ancestor, was exploited by hunter-gatherers both in China and in India. And in China was where believed domestication occurred, where Japonica rice was domesticated in the Yangtze Valley, in the middle Yangtze Valley of China, starting about 9,000 years ago. And this gave rise to Japonica rice, which spread both northwards into Korea and Japan and southwards into tropical Southeast Asia and actually into South Asia as well. Indica was starting to be developed in India, the Ganges area. And here they were either harvesting wild Oryza rufipogon, or maybe an ecotype of it called Oryza nivara, or they were starting to cultivate it, and it was leading to what has been suggested to be a proto-indica. But what happened, we think, is about 4,000 years ago, Japonica made its way into India, probably through the Silk Road, and it hybridized, the Japonica rice hybridized with the proto-indica that was developing in India. And in doing so, it transferred the key domestication alleles from Japonica into Indica, or into this group. And this gave rise to Indica rice, which has subsequently spread westward into into Central Asia and into China and into Southeast Asia. In fact, Indica now is the dominant rice subspecies. I think about 80% of rice production in the world is Indica rice. So that is what we think happened with the domestication of rice. Now, my group right now is focused not on the domestication process itself, but the question of how did rice spread after its domestication? How did it go from being this species that was grown in the Yangtze Valley and spread all around, in this case, Asia for Japonica rice, and well, actually Asia as well for Indica rice. And the evolutionary spread of crops is an interesting topic because it is intertwined with both the migration of Homo sapiens, of us as humans, as we moved around the world, and also cultural interaction, the interaction of different populations of humans as they passed on knowledge of agriculture and passed on varieties that may be useful in different parts of the world. And also in the spread of crops, you have to undertake a variety of adaptations to different climates, different day lengths, different soil environments, different biotic interactions. So the spread of crops actually is an opportunity to study the dispersal of a species, but to study also how adaptation occurs as a species is spreading outside of what is essentially and initially a very narrow distribution range. And so, as I said, for example, in the evolution of rice and the spread of rice throughout Asia, if you look at the climatic zones of Asia, it has to go anywhere from areas that are traditionally thought of as cold climatic zones into temperate and tropical zones, and even possibly into dry, what could even be considered desert zones. And the process by which this occurs also is something that we're interested in. The other reason we're interested in studying the spread of crop species is that, as we'll find out, some of the impetus for the adaptation and spread of the species has to do with responses to past climate change. In this case, for example, these are monsoon records or rainfall records in Asia going back over the last 2,000 years. And we think both rainfall patterns as well as temperature changes are responsible for the adaptation of crops in different parts of the world. And so this is something that we would like to explore. So we started this study several years ago where we took whole genome resequence data for rice. There has been a large genome resequencing effort in domesticated rice. There's about 3,000 rice varieties that have been sequenced throughout the world. This was several years ago. This is called the 3K project. And from this data, we extracted data for about 1,265 land races where they had good geographic information and they had good sequence coverage. In our lab alone, we also sequenced about 178 land races, which we added to this project. We, of course, when you deal with a data set this large, there are several filtering steps you do. Initially, we called 41 million SNPs in this data set, but we filtered in various ways so that in the end, we use anywhere between about, let's say, 500,000 SNPs to maybe 4 million SNPs in various analysis, depending on the analysis we use and depending on what subspecies, whether we looked at Indica or Japonica. And this is just giving you kind of the flow chart for the filtering steps we did to get the SNP set for the analysis. And so we, as I said, we focus our attention on 1,440 plus land races. That was part of the 3K genome data set, as well as some that we sequence in our laboratory. And when you do a multidimensional scaling analysis or a principal component analysis for the data set, you see that rice generally falls into 3 distinct groups. These represent the major subspecies. Here on the upper left, you have the Indica rice. On the upper right, you've got Japonica rice, and here in the bottom, you've got Ausch. The aromatic rices are found here. Our work and others have shown that the aromatic rices like Basmati turn out to be a hybrid between Ausch and Japonica. So these are the major groups of rice from these 1,443 land races. Right now, I'm going to focus our attention on Japonica rice, which is the rice here in the upper right-hand quadrant, and where we have 372 land races for this subspecies. And when you blow up that right-hand quadrant, what you see are you can see distinct genetic clusters even within Japonica for these different rice land races. And in fact, these genetic clusters group very nicely with different geographic regions for which these land races are grown. So in the upper right, you've got temperate rice from Northeast Asia. This is primarily from Korea and Japan and Northeast China. This is the one that gives you sushi, for example. There's another group of Northeast Asian temperate rices that grow in the upland regions. In Southeast Asia, these are a group of rices that are found in the mainland, Laos, Malaysia, Thailand, and so on. And this group here are rice, the Japonica rice, that is found in island Southeast Asia, so in Indonesia and the Philippines. So as you can see, there's nice geographic patterning of these discrete genetic clusters of Japonica rice in the region. So with our identification of these genetic clusters, the question is, how are they related to each other? And by looking at the relationships in concert with archaeological records, can we reconstruct the history of the dispersal of these different rice groups? So we use a process that some of you may be familiar with, used in human population genetics quite a bit, admixture graph analysis, wherein we reconstructed the relationships between these different geographic populations of Japonica rice. And the nice thing about admixture graph analysis is not only gives you the relationships of the geographic populations, but it will also tell you areas where there is evidence of admixture as an ancestor to different geographic subpopulations. And if you take this admixture graph analysis and you also look at archaeological data and the geography of where these land races comes, you can actually develop a model for the dispersal of these different geographic subpopulations of Japonica rice. And not only that, obviously, from population genomic analysis, you are able to provide estimates of the times of divergence of these different major divergences in the dispersal associated with the dispersal of Japonica rice. Now, as you can see, it's really complicated. There's a lot of arrows going all over the place. So what I'd like to do is let's take it one by one and kind of deconstruct what we think is happening in the dispersal of Japonica. So these are where the subpopulations are. The initial thing is we think that there was domestication of Japonica rice starting about 9,000 years ago in the Yangtze Valley, somewhere in this area. Actually, it seems to be too far west. It should be somewhere here where we think rice was initially domesticated. And the first divergence we see in our admixture graph is really a split between temperate and tropical Japonica. The temperate Japonica is the Japonica that eventually makes its way into Japan and Korea and in Northeast China in the north, and tropical Japonica, which splits off and goes into mainland Southeast Asia and to island Southeast Asia. And using cross-coalescence analysis of tropical versus temperate Japonica. And by the way, that split is actually also observed in phylogeny. So if you look at the Japonica group in that analysis I showed you earlier, here's tropical Japonica, here's temperate Japonica, and they are distinct in this phylogeny. We date the split here using cross-coalescence to somewhere between 4,100 to 2,500 years ago. That's when we think that this split started to occur about 4,100 years ago. That gives rise to both temperate and tropical Japonica. And that's interesting because we started to ask ourselves what happened 4,000 years ago that may have put evolutionary pressure to give rise to this divergence between temperate and tropical rice. And the interesting thing is that this period in time is known by paleoclimatologists quite well. This is an area called the 4.2K event. This was a time, if you look at the lower part of this panel, this was a time when globally the Earth started to cool. And actually this cooling event created major disruptions around the world. It caused actually the collapse of some civilizations in the Middle East. It caused turmoil in the developing Chinese empire, the Yellow River, and so on. So this climate event, which is now called a 4.2K event, which is associated with a split of temperate and tropical Japonica, we think is an important event that may have spurred the evolution of these two different subspecies. Here you see the distribution of the cross-coalescence. We see the split starting about 4,100 years ago between temperate and tropical Japonica. Also, if you look at the effective population size in temperate and tropical rice and how it's behaving, we see that there's actually this major bottleneck that starts off right at about the time we think the divergence of temperate and tropical Japonica occurs, and it occurs at this 4.2K event. Now, just to show you dramatically how much the climate changed in Asia during this time, we teamed up with some colleagues who are paleoclimatologists and archaeologists. Jane Guedes at the Scripps Institution of Oceanography, and Kyle Businski at the Crow Canyon Archaeological Center. And what they did was they looked at the requirements for growing degree days for tropical Japonica and asked, where in Asia could tropical Japonica grow? And this shows you in the map 5,500 years ago where tropical Japonica can grow in Asia. It can grow pretty much everywhere except the Tibetan Plateau. And what this is a movie. We're going to go forward in time. And you're going to see, as you see darker colors in this map, these are areas where tropical Japonica no longer is favored as a place where to grow. So I'm going to go with this movie, and you can see about 5,000 years ago, and starting about 4,500 years ago, somewhere in the north, you're no longer able to. Here's the 4.2K event. Oops. So here you can see what's happening. Now the northern part of Asia is no longer a place where tropical Japonica can grow, right? This is now about 3,800 years ago. And this is where Asia is now. Oops, I'm sorry. This is where Asia is now today. So let's run this again. The 4.2K event starts right about now. You see this massive cooling that really makes the north inhospitable for tropical Japonica. And pretty much that's what Asia looks like now, where tropical Japonica can no longer grow pretty much, north of, let's say, the Pearl River in the Guangzhou region. You have to be a more temperate Japonica in order to grow there. Tropical Japonica can only grow now in mainland and island Southeast Asia. If you look at the archaeological record, you actually see this split. Start between 9,000 and 4,200 years ago. If you look at where you can find remains of domesticated rice in Asia, they're found pretty much in the north, in this area between the Yellow River, the Yangtze River and the Yellow River. But after 4,200 years ago, Japonica really bifurcates, where you see it growing in Northeast Asia and in Southeast Asia, in mainland Southeast Asia. And later on, you're going to see it in island Southeast Asia. So that's what we think is the major split in the evolution of Japonica. And subsequent to that, we see other splits. So after the split of temperate and tropical Japonica, we suddenly see a split between mainland and island Southeast Asian land races. And also within island Southeast Asia, we see colonization of, say, the Indonesian archipelago, Borneo, and the Philippines. And when you date the split between mainland and island Southeast Asia, again, using SMC++, losing cross-coalescence, it's about between 2,500 and 1,600 years ago. And within island Southeast Asia, it's about the same time. So when Japonica rice moves into tropical Southeast Asia, almost immediately it starts to diversify into different islands and different parts of the mainland, much more recently than the domestication of Asian rice. Now, when we were looking at this, and this was published, we published this in Nature Plants last year, you can read about it. But one of the things that puzzled us was the relationship between the origin of the rice in island Southeast Asia. Because the origin of rice agriculture in island Southeast Asia has been contentious over the last 2 decades. There was a major theory that was proposed that rice agriculture made it into island Southeast Asia as a result of the Austronesian expansion starting about 4,000 years ago. So as many of you may know, the Austronesians, which now are the ones who populate much of Polynesia and have impacted the culture of Southeast Asia, of island Southeast Asia especially, is believed to have originated from indigenous peoples of Taiwan starting 4,000 years ago, where they moved out of Taiwan and they spread throughout island Southeast Asia and into Polynesia. And it's thought that they brought rice agriculture with them. At least Peter Bellwood, who was the one proponent of what we call the out of Taiwan hypothesis, was that rice agriculture came to island Southeast Asia from Taiwan. But if you look at our analysis, it seems to have come from mainland Southeast Asia. The problem is our sampling in Taiwan, actually, and parts of the Philippines was not very robust for this. So we collaborated with a colleague of mine, Caroline Singh, and by the way, this idea of the Austronesian expansion, there was a recent paper that was published just 3 weeks ago, which suggested that the peopling of Southeast Asia, island Southeast Asia, may be more complicated than this out of Taiwan hypothesis. It's certainly true that Taiwan and certain groups in the Philippines, especially in the northern island of Luzon, may have been populated essentially originally from mainland China into Taiwan, but separately into the Philippines as well. But many of the Philippine groups came from the south, from mainland Southeast Asia, and much earlier than the Austronesian expansion. So this was a nice paper in PNAS a few weeks ago, last month. What this suggested also is that the rice agriculture itself may have been separate from the Austronesian expansion. So what we did with Caroline, our collaborator from Academia Sinica in Taiwan, is she had sequenced varieties from the indigenous peoples of Taiwan. These are grown in the mountains of Taiwan. These are not the Han Chinese immigrants into Taiwan that came in much later, but represented rice that was grown by indigenous peoples. And we also had varieties sequence, landraces sequence from the famed rice terraces of northern Luzon, which are also farmed by the indigenous peoples of northern Luzon, the people of the Cordillera region. And so what we did, and this is now under review in MBEs, we incorporated that in our analysis. And when we do the admixture graph, now including these new Taiwanese samples and these Philippine samples, we see pretty much the same picture, except now you see these 2 Taiwanese samples. One Taiwanese population, groups close to mainland Southeast Asia, another groups close to island Southeast Asia. The northern Philippines group is really found in the middle of island Southeast Asian groups from other land races in the Philippines and Indonesia. And if you look at an admixture graph analysis, like we did in the previous one, what you see is pretty much, it allows us to replicate what we had in our previous analysis, but then put in the Taiwan and northern Philippines into this analysis. So here you see the temperate tropical Japonica split, you see the mainland and island Southeast Asia split, and here you see the diversification in island Southeast Asia. These are the splits we find. But if you look at the Taiwanese land races here, you see an interesting and more complicated picture. So look at Taiwan 1, this population here. What it appears to be, an admixture between a temperate population that is closely related to the Northeast Asian lowland temperates you find in Korea, China, and Japan, and from the northern Philippines, from the Cordillera region in the Philippines. So remember, it was thought that maybe the northern Philippine land races originated from Taiwan. But here we're seeing the reverse. If anything, we're getting gene flow from the northern Philippines into Taiwan. And Taiwan originally is a temperate variety with admixture from the northern Philippines. The other one, Taiwan 2, has the same picture. You have a admixture from what's a temperate lineage in Taiwan, and the admixture comes from mainland Southeast Asia. Well, here we have Bhutan, but it's really from Laos, which itself is a complicated admixed population. And so we see a picture here where these Taiwanese population, rather than being donors or being the ancestors of populations in island Southeast Asia, they're actually recipients of gene flow from Southeast Asian populations. We've used DADI analysis to look more closely at the parameters, both for the migration and the timing of the divergences we see here. These are the results of DADI. The temperate population that colonized Taiwan from Northeast Asia, from Korea and Japan and Northeast China, we think these two diverge about 2,600 years ago. And here you can see this divergence about 2,600 years ago. And here we see, in this one, we can see that definitely you're getting gene flow from the northern Philippines into Taiwan. There's a little bit from Taiwan into the northern Philippines, but definitely it's overwhelmingly from the northern Philippines into Taiwan. And so the picture we are seeing here is after the split of temperate and tropical Japonica about 2,600 years ago, a population made it from the temperate split into Taiwan. And more recently, and we're not about 1,300 years ago, or actually greater than 1,300 years ago, there was gene flow from the northern Philippines into Taiwan itself. And the thing about this is that during this time period, 2,500, 1,000 to 1,500 years ago, there was extensive trading in this region between China, Taiwan, and the islands and mainland of Southeast Asia. This entire region has been thought of as the Austronesian trading sphere. And we can see things like jade items, which were originating in Taiwan and spreading all throughout island Southeast Asia. The material is coming from Taiwan, but itinerant jade craftsmen are spreading it all over Southeast Asia. And also from North Vietnam, there are bronze drums that begin to be found in island Southeast Asia, starting about 2,400 years ago. So this area of the world is not static. It is the center of a large trading sphere where we think they're moving not only material goods like jade and drums, but maybe they're also moving rice seed that's leading to the movement of rice around this area as well. So we think that's what's happening there. Now, mainland Southeast Asia, the split in island Southeast Asia from mainland Southeast Asia is also interesting. When we look at our data, say between the northern Philippines and island Southeast Asia, we're seeing a divergence of about 3,400 years. Here's between mainland Southeast Asia and Indonesia. This is actually between Indonesia and the northern Philippines. It's about 3,400, 3,500 years ago using DADI analysis. Now, remember in our previous analysis where we used cross-coalescence, we dated those splits at about 2,500 years ago. Here we're seeing a date that's about 1,000 years earlier. And we still have to resolve this discrepancy. I think we need more sampling to try to, and more analysis to resolve whether it's 2,500 to 3,500 years ago. But certainly during this time period is when we see the diversification of rice in Japonica, sometime during this area. And now this is, again, if you look at the past climate, this is an interesting area. In Southeast Asia, actually, starting about 5,000 years ago to 3,200 years ago, there was a massive drought that's called the mid-Holocene drought, or the Southeast Asian megadrop, where there was a dramatic reduction in precipitation and the monsoon was very weak during this period. It started to recover about 3,200 years ago. And this time period has been called the missing millennia in Southeast Asia, because what you see is prior to that, you see hunter-gatherers. But during the missing millennia, you don't really see much in terms of agriculture. It's only after about 3,200 years ago you see cereal crop cultivators in this region. So we think, again, this is purely because of the overlaps in the time. It may be that prior to 3,200 years ago, you are not able to grow rice in Southeast Asia to any extent because of this drought. It's only later on that you're able to get rice into Southeast Asia. So here we're able to with our work, we're able to start to reconstruct both the patterning of the dispersal of Japonica rice in Asia, and also aspects of the timing of this dispersal. And as you can see, we can correlate this with either past climate events or with even cultural artifacts and their movement in this region. Now, the Indica rice, which is now the predominant rice subspecies, is an interesting question. And it turns out to be much more complicated to look at Indica rice than it was to look at Japonica rice. So we're able to do the same thing. We're able to look at admixture graphs and to reconstruct it. But our admixture graph reconstruction for Indica is actually fairly unstable. It's very unstable depending on what populations you put in and how many discrete populations you specify in the model. And because of this, our understanding of how Indica spread into China and to Southeast Asia is much less clear. The one thing that we can say for sure is that there was an early split between rice that went Indica that was in South Asia or India, Sri Lanka, and ones that went to China and later on to Southeast Asia. So we think that, of course, Indica rice was domesticated 4,500 years ago, and the split between India versus China, Southeast Asia occurred much more recently, between 2,000 and 1,400 years ago, based again on our cross-coalescence estimates. This is, again, we love to look at for what we think are correlations. Could be coincidence, maybe it's a correlation. This is an area in time when there's cultural interaction between China and India. Buddhism moves from India into China at about this time. This was, we can date exactly when Buddhism moved to China because we know who brought it into China. But this actually shows you the amount of strong cultural interaction between South Asia and East Asia at this time. And also, in central Thailand, it's about 1,800 years ago, when you begin to see Indica rice in the archaeological record show up in central Thailand, again suggesting that our dating of the movement of rice from South Asia into China and Southeast Asia is concordant with what we know about cultural interactions and also what we know about the archaeological record. So that gives you what we know about Indica. There's still much work that needs to be done. As I said, one of the directions we're going here is we want to understand local adaptation associated with the movement of rice land races in this region. We've started to do that with our analysis of the Taiwanese and Northern Philippines samples using a method called the graph-aware reconstruction of selective sweeps. We're able to identify, for example, 2 gene regions, one that's associated with actually UV tolerance. These are, by the way, Northern Philippines and Taiwan 1 rices are grown in the mountainous regions. And then another that's involved in what we might think of as biotic interaction. So this is an ongoing area of investigation where we're trying to understand selection associated with the movement of rice in different rice populations. So to summarize this part, we think that dispersal and diversification of Japonica rice, especially the initial evolution of temperate and tropical groups of Japonica rice, is associated with this climate change event 4,200 years ago. In Southeast Asia, it's associated with the end of the Holocene megadrop 3,200 years ago. And we think now we have evidence that Japonica rice in Southeast Asia is not associated with the Austronesian expansion from Taiwan. The idea that rice agriculture is associated with the Austronesian expansion in island and mainland Southeast Asia has actually been questioned very strongly by archaeologists who don't see much evidence of it in the archaeological record. And now we're beginning to see that the genomic data is beginning to support this idea that rice agriculture in Southeast Asia was brought independently from Taiwan, coming from Taiwan. So the future directions here, there are many. We're continuing this project quite heavily. One is that many of the land races we have in our analysis do not fall into these discrete clusters, and so we don't include them in the analysis. We think there's a lot of information there. We need to think more carefully how to look at admixed individuals and how they can contribute to our understanding of dispersal. As I said, we want to study selection and genes associated with this dispersal. Some of our sampling is not very complete in China for Japonica, for example. There's a lack of land races because Indica pretty much replaced Japonica in China. So, and also parts of mainland Southeast Asia. So what we've done here is we've gone to herbarium specimens where they've collected rice land races earlier than the 1950s up to like the 19th century. And we've actually already extracted DNA from about 100 Japonica land races in mainland South, or we're in the process of doing that in mainland Southeast Asia and China. And then hopefully that make complete our picture of Japonica dispersal. And finally, you know, Indica and the other subspecies, Aushan basmati, we're working on trying to reconstruct their dispersal. We still need more work in that area as well. So there's still a lot to do in this area that we're excited about. And by the way, in terms of rice projects, the spread of rice projects is one of several projects we've been doing on the evolution of rice in my lab. We have looking at selection on gene expression. This is a project that we did that we published in Nature last year, and we're continuing to do that. We're interested in how gene expressions is selected for, especially in the adaptation of rice to different agricultural ecosystems. We have the Rice Fitness Consequence Map project where we looked at, we are able to map regions of the rice genome, especially non-coding regions that may have evidence of strong selection. And we also do a lot of systems biology where we reconstruct gene regulatory networks and look for adaptations of genes associated with different environments in the context of their genetic network. So the rice projects in the lab are not just the spread of rice, but I thought that's what I would talk to you today about. Very quickly, for the next 10 or 15 minutes, I'd like to switch to another project looking at date palms, or Phoenix dactylifera, which is another important model system we started to work in our lab. And this is work that we do in the United Arab Emirates and Abu Dhabi. Now, NYU, some of you may not know this, that New York University is actually a global university. The name, of course, says that New York University is centered in New York City. That's where it was founded, and that's where our biggest campus is. But we have actually two other campuses in the world where we grant degrees. One is in Abu Dhabi, the United Arab Emirates, NYU Abu Dhabi, and NYU Shanghai. And these are major degree granting campuses of New York University. The idea of New York University is we're a global university, so you know, faculty and students can move into different campuses. So for myself, for example, I have laboratories in New York City and Abu Dhabi. And about 10 years ago now, 10 years ago now, we, you know, as we were founding the New York Abu Dhabi campus, me and several colleagues in New York decided to establish the Center for Genomics and Systems Biology in Abu Dhabi that mirrored the one that we had in New York City. And when I started to do work in Abu Dhabi, you know, rice is not a good crop in Abu Dhabi, even though it's actually a major part of their cuisine. It's in the desert. But looking around, we started to get interested in date palms. And for several reasons. One, it's the indigenous crop of the Arabian Peninsula. So it seemed like an appropriate crop to study in the United Arab Emirates. But the other thing is that, you know, I've been working in rice and other annual crops, and they had their particular evolutionary dynamic. But perennial tree crops or perennial crops have a different evolutionary dynamic. And I know that Brandon Gott gave a talk a few weeks ago and talking about grapes, which are also another perennial fruit crop. And it's interesting to look at perennial crops and how they relate to annual crops in terms of their evolution. So there's a dactylifera genome. It's about 850 megabases. Last year we released, was it last year or the year before? We did a PacBio sequencing where we have a really good assembly of the genome. There are 18 chromosomes. It's a dioecious tree, so it's got separate male and female trees. And there are about 3,000 varieties, it's thought, of date palms. Date palms, unlike rice, there is no scientific breeding in date palms. All of them are traditional land races. There might be a few that have been developed in California, but really these are traditional varieties that are available throughout the world. When you look at where Phoenix dactylifera is grown, it's grown anywhere from, you know, Pakistan and parts of India into the Arabian Peninsula and the Fertile Crescent and into North Africa. That's really where the traditional range of date palms are. In modern times, it's also grown in Australia, it's grown in Mexico, and it's grown in the United States as well. But traditionally, this is where date palms are found. And what is thought is that date palms originated somewhere in the Gulf region or the Fertile Crescent, but probably the Gulf region, and from there spread eastwards into Pakistan and India and westwards into North Africa. And we think it started to be domesticated maybe 7,000 years ago. There's certainly archaeological evidence for at least pre-cultivation exploitation in Dalma Island. But by 5,000 years ago, there's already written descriptions of plantations of date palms in the Mesopotamian area, the Fertile Crescent. So somewhere about that time when it was domesticated. One of the things that's interesting in date palms is that we don't know what the ancestor is. The sister species is Phoenix sylvestris, which is found in India. But for a variety of reasons, that's not thought to be the ancestor. So there's still a kind of a hunt for what is the ancestral population that gave rise to cultivated domesticated date palms. That's still an area of active research. There is quite a bit of diversity. If you are from Europe or the United States and you have a date, you're likely eating just one of two varieties, either Deglet Noor or Medjool, which are primarily from North Africa. But if you go to the Middle East, as some of you go, if you go to Dubai or Kuwait or any place in the Middle East or Cairo, and you go to the market, you will have a lot of varieties to choose from. And they differ in sweetness, they differ in size, they differ in color, they differ in their, subtle aspects of their taste and what they're used for. And so it's this diversity we're interested in looking at. And one of the things we did several years ago now, where we started to sequence the genome of date palms across the range. This is a sample of 60 date palms. It was clear to us that genetically, the Middle East date palms were distinct from the Northeast date palms. Here's a structure analysis where you can see the Middle East date palms, like one made primarily from one source, the North African date palms from another source, and the ones from Egypt and Sudan look like they're a hybrid between the two. This suggested, of course, that maybe there is separate domestications of these, but we thought there might be a better way to think about this. So if you look at the archaeological record, earliest ones are in the Gulf. Then you see about 5,000 years ago in the Levant, then about 3,500 years ago it goes into Egypt, 3,000 to 2,000 years ago it's in North Africa. And so this suggests, right, if you look at the archaeological record, what we're seeing is a movement of dates from the east to the west. And as we would expect, because we would expect this might be from serial founder events, the greatest diversity of dates would be found in the Middle East. And North Africa, you would have reduced diversity. However, we didn't see that. If you actually look at theta or pi, theta, for example, the North African varieties are about 20% more diverse than the Middle East varieties, which was counter to what we thought about in a kind of a serial founder population model going from east to west. But one of the things we decided in order to look at this more closely was we decided to sequence samples from other wild phoenix species. The genus Phoenix has 13 species, and many of these are somewhat sympatric or parapatric with Phoenix dactylifera. And we sequenced several samples of those. And what we found, to our surprise several years ago, was if you look at North African date palms, they look like they're a hybrid between Middle East Phoenix dactylifera and the species Phoenix theophrasti. And Phoenix theophrasti is an interesting species. So if you look at the phylogeny, this is date palms, Phoenix dactylifera. This is sylvestris, which is the closest species to Phoenix dactylifera. Phoenix theophrasti is somewhat more distant from Phoenix sylvestris. You have other species here. We rooted here with Phoenix franklinana, which is found in sub-Saharan Africa. So it appears, as I said, that North African date palms are a hybrid between Phoenix dactylifera and Phoenix theophrasti. And what's Phoenix theophrasti? Phoenix theophrasti is a date palm that right now is found only in the island of Crete, a few of the other Greek islands, and in southwest Turkey. If you look at Crete right now, in Crete, you have the largest palm forest in Europe, and it is all Phoenix theophrasti. This is the island resort of Vai in Crete. And you see Phoenix theophrasti here growing. If you look, you can even just do Google Maps of the island of Crete, and you can find Phoenix theophrasti. This is in, I think this is in, this is in Vai. This is what I showed you earlier, next to this beach resort. Here's Prevely Beach, and here's Phoenix theophrasti growing. There are about 10 populations of Phoenix theophrasti in Crete. And as I said, we think what we're seeing here is this is the source species that hybridized with Phoenix dactylifera and gave rise to North African date palms. And there are many ways to test it. It was not just a structure graph. We did patterns. We had the Abba-Baba test. We have F3 and F4 tests. All of them are consistent with this idea that North African date palms are a hybrid between Theophrasti and Middle East Phoenix dactylifera. Tree mix analysis shows you hybridization between Theophrasti into North Africa, which is the North African more related to the Middle East. And if you look across the genomes, you are able to find haplotypes, fairly large haplotypes, wherein they are identical between North African date palms and Phoenix theophrasti from Crete. Again, showing you there are these regions of the genome that show introgression from Phoenix theophrasti into the North African date palms. And so the picture we have now is date palms we think originated 7,000 years ago. Then it spread throughout the Middle East, and then it hybridized with Phoenix theophrasti. We don't know particularly where. And then that hybridization gave rise to the North African dates that spread westward into North Africa. And if you look at the artifacts in Crete, for example, you see that palms are an important part of their culture. These are the Minoan this one is from the Minoan civilization in Crete. You see dates in palms in their pottery. The coinage here much later, you see date palms. This is clearly a date palm in Crete 2,300 years ago. During this period, there of course was a lot of movement in this region. The Minoans and later on the Phoenicians, more famously, were traders that traded all across the Eastern Mediterranean into the Western Mediterranean. There's movement of cultures of trade goods in this region. It suggests also there might have been movement of biological material. Where in this region the hybridization occurred, we do not know. But certainly it's not implausible to think that with the movement of peoples and trading in this area, there were opportunities for Phoenix theophrasti here in the Aegean Islands in southwest Turkey to hybridize with Phoenix dactylifera in North Africa. And by the way, we don't know 3,000 years ago, Phoenix theophrasti may have also populated the Levant and North Africa. We don't know that. There's one example in Israel where there's believed to be a Phoenix theophrasti seed in the archaeological record. There's just one seed, so we don't know what that means. Now, I'll tell you very finally in the last 3 minutes, this is coming out in press right now. In order to look at the dynamics of this introgression of Phoenix theophrasti, we were able to take advantage of a unique population of date palms in the southern Levant, modern-day Israel, Palestine, and Jordan. And here there are archaeological sites dating from about 2,200 years ago, where there were date palm seeds that were recovered from Wadi Makut, from Qumran and Masada. And a colleague of mine, Sarah Salon and Elaine Salawi in Israel, took these date palm seeds that were 2,000 years old, and they managed to germinate them. So they first germinated the one from Masada, which they called Methuselah. They germinated those in 2008, and later on they were able to they planted 35 other archaeological date palm seeds, and a total of 7 actually germinated and are growing trees right now. So there's Methuselah, there's Hannah, and they've been named with different names in them. And so we were able to sequence these date palm genomes. And what we found was interesting. These different seeds were dated from, the earliest is Methuselah, which dates sometime between the 4th century, the last 4 centuries of the Common Era. And Hannah is about the same age. Adam and Judith are slightly later. And then Boaz, Jonah, and Uzziel are likely later. Uzziel and Jonah and Boaz are probably now into the Common Era, 1,800 to 2,000 years ago. So there's a nice age progression of these seed. So we sequence the genomes, and what we find is really interesting. The two oldest genomes, the two oldest seeds, Methuselah and Hannah, actually look like they're Middle East dates, or they're from West Asia, while the other four look like they're more North African dates. So you see a transformation of the dates in this region going from more Middle Eastern varieties into more North African varieties. And the heterozygosity of these dates from the Levant are actually more similar to North African dates than they are to West Asian dates. The other thing that's interesting is we actually begin to see introgression. And the Methuselah, the oldest date seed, we don't see evidence of Phoenix Theophrasti. In Hannah, we see a little bit of Phoenix Theophrasti in the genome, but certainly after Hannah, starting about 2,200 years ago, we start to see Theophrasti in the genome, and the level of Theophrasti introgression into these dates increasing with as time progresses. We also see that in these ancient samples, we painted the chromosomes and the track lengths of Phoenix Theophrasti, the introgression lengths, are larger in average than even the introgression lengths in North Africa, suggesting, of course, that this may be, since this is closer to the hybridization events, we are still seeing larger non-recombined introgression tracts of Phoenix Theophrasti into Phoenix dactylifera. And you can see so very nicely as time progresses in this region of the southern Levant, you see increasing heterozygosity, you see an increasing switch of Middle East to North African dates, and you see increasing levels of introgression from Phoenix Theophrasti. But certainly by 2,200 years ago, you now see Phoenix Theophrasti introgression into Phoenix dactylifera. So here, very simple West Asian, you get into the southern Levant, you get Phoenix dactylifera going into the region, but later on, you're seeing North African Phoenix dactylifera with Phoenix Theophrasti introgression moving into the area as well. By the way, it's moving into this area as well with the rise of the Roman Empire and the movement of the Roman Empire into this region, where, of course, they're probably moving goods and material from their colonies in North Africa into the southern Levant. So we think date palms likely originated in the Raven Gulf. There is evidence of hybridization with Phoenix Theophrasti of these migrating date palms, and these form the North African dates. We think this hybridization had already occurred by 2,200 years ago. And in the southern Levant, we see the impact of North African genotypes over time from the 4th century before the Common Era to the 2nd century in the Common Era. In the future direction here, so we're actually now analyzing a very large data set here, about 1,000 varieties. We're continuing to see the role of Phoenix Theophrasti into North African date palms, and we have evidence of another date palm wild species, Canariensis, in the Canary Islands, that may have also impacted the evolution of date palms, and we're also looking at genome evolution in genus Phoenix. And I'm sorry I went a little over time, but as you can see, this is an exciting area where we're now trying to reconstruct the dispersal of species, domesticated species after their domestication. And we think this tells us something about adaptation of these species as they move around the world. I'd like to thank several people for the rice work. The talk I discussed really the talk mainly of Rafal Gutaker and Ornob alum in my lab. Rafal is now the Kew Gardens. He was a postdoc in my lab. Ornob is a graduate student in my lab. He did the Taiwan Northern Philippine study, and other people in the lab contributed to our understanding of the spread of and the evolution of rice in our work. In the date palm project, the work I showed you was Jonathan Flowers and Muriel Gros-Balthasard. Muriel is now back in IRD in Montpellier in France, and other people in my lab in Abu Dhabi are working on this. And this was funded by several funding sources, the Ziegler Family Foundation, the National Science Foundation, and NYU Abu Dhabi. And with that, I'd like to thank you for your attention. I'm sorry I went a little bit over, and I'm happy to take any questions if there are.